Research Organization for Health, Research Organization for Nanotechnology and Material, Research Organization for Agriculture and Food, Research Organization for Governance, Economy and Community Welfare. BRIN catalyzing research and innovation to develop an advanced scientific ecosystem in Indonesia with two main targets. BRIN realizes the tremendous potential of the nation's youth in the field of science and technology. Thus, various research and innovation policies are being formulated to develop and cultivate human resources with high competence and aptitude in science and technology. Creating many excellent programs is a form of BRIN's commitment to researchers, innovators, and the general population from various disciplines across the nation. The research and innovation ecosystem quality improvement in Indonesia is also realized through the collaboration of various ministries, institutions, domestic industries, and regional governments, to name a few, by forming the Regional Research and Innovation Agency, or RIDA. Moreover, cooperation with various foreign research institutions continue to be developed through active participation as member of various organizations and international forums. BRIN organizes research into a more effective and efficient process by integrating human resources, infrastructure, and budget from various research and development institutions. Oriented by a national research priority, BRIN routinely enacts comprehensive evaluations from various research and development institutions. This effort is supported through the continued management and improvement of various research and innovation facilities located across 17 regions of Indonesia, dedicated towards the development of science and technology. BRIN continues to improve our contribution to research and innovation towards national development by developing and revitalizing 4 areas of science and technology, or KST. An integrated area consisting of research laboratory facilities, testing laboratories, and industrial facilities for both startups and SMEs, tenant office rooms, co-working space, meeting rooms, gallery rooms, and other supporting facilities. BRIN is here to manifest the future of a golden Indonesia 2045 by consolidating science and technology resources and creating a resource ecosystem and research-based economical foundation. Transforming Indonesia as a global reference in the development and application of science and technology, practices, and the good of humankind. Nature is the source of inspiration. Knowledge is the foundation, and to be put in praxis for the sake of the nation.
Kenapa kok ini?
Okay, good morning, everyone. I welcome all today. Welcome to today's event entitled The Two Phases of Bilirubin, from Neonatal Toxicity to the Potential Protection in Neurodegenerative Diseases. This event is conducted as a part of the ongoing research and educational collaboration between National Research and Innovation Agency of Indonesia, or known as BRIN, with the Fondazione Italiana Fegato Onlus, or known as FIF. My name is Naning, and I will be your host for today. Before we begin, I would like to take a moment to welcome all of you, distinguished guests, our excellent speakers, and also online participants. This webinar objective is to explore the management of bilirubin toxicity as well as the potential role of bilirubin in neuroprotection. We have a distinguished panel of speakers who will share their expertise on various aspects of these topics. And here's a quick overview of today's agenda. Firstly, there's opening remarks by Professor Dr. NLP Indi Narmayanti, MSc, as the chairman of the Research Organization of Health, BRIN, continued by Professor Claudio Teribelli, MD, PhD, the scientific director of FAIF, and lastly by Elizabeth Paranovita Kotrier, PhD, as the head of Ekman Research Center for Molecular Biology. And following the opening remarks, there will be a presentation on our today's topic on the potential role of bilirubin in neuroprotection that will be delivered firstly by Dr. Sylvia Gatsin, PhD. She is the senior researcher at FIF, and then followed by Dr. John Polido, the PhD candidate from the University of Trieste, Italy. And lastly, the presentation by the postdoctoral researcher at the Ekman Research Center for Molecular Biology, Sri Jayanti, MD, PhD. And without further ado, please welcome Prof. Indi to deliver the opening speech. And because Prof. Indi doesn't join us online, we have prepared the video of the opening remarks. Okay.
Speakers, colleagues, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. First, let us express our gratitude to the presence of Allah Subhanahu wa Ta'ala that we are in good health and we can be here to attend this webinar. On behalf of Research Organization for Health, National Research and Innovation Agency of Indonesia, I would like to welcome you in this webinar entitled The Two Phases of Bilirubin from Neonatal Toxicity to the Potential Protection in Neurodegenerative Diseases. This webinar is conducted as part of the ongoing research and educational collaboration between the National Research of Innovation Agency of Indonesia with Fondazione Italiana Fegato ONLUS, signed in Trieste, Italy, 28 September 2022. Ladies and gentlemen, the level of bilirubin is one important issue in neonates. Chronic thirst arises as a hazard consequence of severe neonatal hyperbilirubinemia, wherein exercise and conjugated bilirubin traverses the blood-brain barrier, causing irreversible harm to the brain cells. This condition remains a significant issue globally, particularly in developing nations with a limited medical infrastructure. In Indonesia, it has been reported that approximately 7% of infants experience a severe hyperbilirubinemia. Additionally, acute bilirubin encephalopathy was diagnosed in 2% of the newborn survived. Exploring the management of bilirubin toxicity as well as the potential role of bilirubin in neuroprotection would provide valuable insight for researchers, clinicians, and policymakers in Indonesia to improve both neonatal and elderly health outcomes. Ladies and gentlemen, on the other hand, besides being considered as harmful molecule in neonates, a small amount of bilirubin is now recognized for its therapeutic potential. It condition like Gilbert syndrome, mildly elevated bilirubin level may offer protection against cardiovascular diseases, cancer, and metabolic syndrome. Bilirubin shows antioxidant and anti-inflammatory properties and is a link of various health conditions, including neurodegenerative diseases. However, research on its protective effect in neurodegenerative diseases like Parkinson's disease is limited. Neonatal jaundice remains a significant issue, and the rising prevalence of neurological disorder like Parkinson's disease. Indonesia faces an increasing burden of disability and healthcare costs. Through the webinar on these topics, we hope we widen the network to bilirubin studies and open a chance for research collaborations. Eventually, we hope we contribute to global health effort to reduce morbidity and mortality associated with these conditions. Hopefully, this webinar can broaden insight and exchange knowledge for the participants, especially those involved in health research, and hopefully hepatitis researchers will gain valuable information. I hope the webinar will run smoothly with the maximum impacts. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Prof. Indi. And I would like to invite Professor Tiribelli to deliver the opening remarks for today's webinar. Buongiorno, Prof. Yeah, Prof. Tiribelli, I think you're still on mute. Yes.
Okay. Good morning to you. Saliman pagi for us. Saliman siang for you. I mean, let me tell you that I'm really thrilled to be here today because this is the nicest demonstration how the cooperation can go on and actually be the bridge between two different countries linked by science. The issue of bilirubin is one important aspect of our life because bilirubin has been considered till 20 years ago as sort of a waste product, is the garbage, the end story of the heme metabolism. Since 20 years, we started understanding that bilirubin may have a lot of different interaction within the cells. And now the concept of bilirubin as a yellow hormone has been solidified. And now we know that bilirubin is able to interact with a lot of different metabolic steps in the cell and in the body. And therefore, bilirubin may be something which can help us to keep our health strong. This is true when the level of bilirubin is slightly higher, as in Gilbert syndrome, and when our age is older than, let's say, 3 days. On the contrary, when we are newborn and the bilirubin level is increased, bilirubin can really be harmful for our health and can cripple the rest of our life. This is why the surveillance of bilirubin metabolism in the first hours of our life is so important. And let me tell you that I will be happy and pleased to revitalize a program which actually started more than 10 years ago in Indonesia to monitor the neonatal jaundice in several different locations of the country. And this is because we know now that the genetic background may play a major role in modulating the bilirubin level. So I think that if together with BRIN and together with the Ministry of Health of Indonesia, we can get together a team in devising a study, a nationwide study in different locations of the country on the bilirubin surveillance level in the newborns from 0 to 72 hours. This is going to be a major breakthrough in the health schemes of Indonesia and the rest of the world. FIF is doing something very similar in the Philippines with good results, but I think that Indonesia has all the assets and all the brains and all the human capabilities to do so. And if this may be put forward, we'll be happy and pleased and honored to help you and to guide you and to interact with you in this program. So I launched this message, and I think that there are several players around this screen able to take over and try to finalize this program. I think this is going to be a major breakthrough. So today we're going to see the two sides of the coins, of the two edges of the sword. The good one, which is dealing with the more advanced age of our life, and this 3 will tell us how bilirubin may be good for preventing some neurological disease. On the contrary, in the first days and the first hours of our life, bilirubin may be dangerous. And Jean-Paul and Sylvia will tell us how we are dealing with this growing health issue. Mind that the neonatal damage done by bilirubin is fully preventable. So if we can manage to do an early diagnosis, we can really wipe out this plague. And I think at this time, it's really poorly acceptable that a newborn may be damaged for the rest of his or her life simply because the diagnosis has been too late. So I think, again, that we need to study this possibility, to explore this possibility, and for sure, I'll be happy and pleased, together with all my collaborators, to help you try to devise a program for preventing severe neonatal jaundice in Indonesia. So I'm not going to remove more time to this symposium, and I pass the podium to our conductor and Nanning, and that's it. Thank you very much.
Thank you very much, Prof, for the beautiful message. And next, I would like to invite Ibu Farah to deliver her opening remarks. Ibu Farah.
Thank you, Nani.
Yes.
Assalamualaikum warahmatullahi wabarakatuh. Good morning to all our participants from Italy, and also good afternoon to all speakers, colleagues, ladies and gentlemen. I would like to thank you, Professor Dr. Hewan NLP Indi Darmayanti, MSc, the Chairman of the Research Organization for Health, National Research and Innovation Agency, BRIN, and also grazie, Professor Claudio Tiribelli, MD, PhD, the Scientific Director of Fondazione Italiana Fegato, for the very inspiring opening remarks. Also, I would like to thank all speakers of this webinar, which entitled The Two Phases of Bilirubin, from neonatal toxicity to the potential protection in neurodegenerative diseases. Thank you, Dr. Sylvia Gatzin, PhD, a senior researcher from the Liver Brain Unit Rita Moretti, Fondazione Italiana Fegato, the Italian Liver Foundation. Also, thank you, Dr. John Paolito, a PhD candidate from the Liver Brain Unit Rita Moretti, the Italian Liver Foundation, University of Trieste. Also, last but not least, I would like to thank Dr. Sri Jayanti, MD, PhD, the postdoctoral researcher of the Eichmann Research Center for Molecular Biology. This webinar is conducted as part of the ongoing research and educational collaboration between the Eichmann Research Center for Molecular Biology, Research Organization for Health, with Fondazione Italiana Fegato, on which were signed in Trieste, Italy, in September 2022. Ladies and gentlemen, we are quite familiar with the fact that some newborns can suffer from jaundice, which also known as the neonatal hyperbilirubinemia, the condition when the total serum bilirubin rises above the normal zone during the first week of life. However, the recognition of bilirubin for its therapeutic potential is quite new. Therefore, we hope that the presentations and discussion of this topic in our webinar today will increase our knowledge. Also, as mentioned by Professor Indi and Professor Claudio, the aim of this webinar is to increase knowledge regarding the importance of treating the neonatal hyperbilirubinemia and Parkinson disease, to introduce the latest research on the pathomechanism of bilirubin neurotoxicity, and new therapeutic approaches for neonatal jaundice, to disseminate the information regarding the results of the translational research, and also to finally to describe the neuroprotective effects of bilirubin in neurodegenerative diseases, especially in Parkinson's disease. So through this webinar, we hope that we can enlarge the network of bilirubin studies and the research collaborations that we have started with the Liver Brain Unit Rita Moretti of the Italian Liver Foundation. And then we hope to expand those collaborations with other universities, hospitals, and institutions. And the last hope, but not least, the outputs of the research, we hope that can contribute to the global health in reducing the morbidity and mortality associated with bilirubin and neurodegenerative diseases. With that, I would like to say thank you and enjoy the talk. Grazie mille. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ibu Farah. And before we proceed to our main agenda for today, I would like to invite all of you, the participants, to have a group pictures. And therefore, I would like to ask you guys to open the video because I will take a screenshot. Please, everyone, open your camera and I will take pictures. I will count. There is 2 pages. Okay, 1, 2, 3, the first page. Smile. Okay, next one. Next is 1, 2, 3, smile. Okay, thank you, everyone. And I will deliver this session, the main agenda for today, the talk by our excellent speakers. Therefore, I will handling this session to our moderator, Dr. Kori Alkobar. Are you there?
Yes, I'm here, Nani.
Okay, please.
Okay, thank you, our beloved MC. Good afternoon, everyone. Good morning, those of you in Italy. The Honorables, Head of Eichmann Research Center for Molecular Biology, Ibu Farah Kutri, PhD, the Scientific Director of the Fondazione Liver Fegato, Prof. Claudio Tiribelli, our distinguished speakers, our participants, ladies and gentlemen, welcome. My name is Corey, and I will be your moderator for today's. We welcome you again to this webinar on the 2 phases of bilirubin, from neonatal toxicity to the potential protection in neurodegenerative diseases, jointly hosted by the Eichmann Research Center for Molecular Biology BRIN and the Italian Liver Foundation. As we know, bilirubin is a product of catabolic breakdown of the red blood cells, and elevated levels of bilirubin or hyperbilirubinemia may indicate certain disease related to the hepatic and biliary systems, and also has been associated with neonatal developmental disorders and progressive diseases. The topics on today's webinar will explore the management of bilirubin toxicity and the potential role of bilirubin as neuroprotector agent in different clinical settings, and how this knowledge can provide valuable insights for management of bilirubin-associated diseases in neonates and adults. Before we start the presentation, I just want to share some housekeeping rules. So we have 3 speakers today, and I will briefly introduce each of our speakers before their presentation started. I will also issue a 2 minutes reminder before the end of allotted slots for each speakers in the interest of keeping time. And the Q&A sessions will be held after all 3 presentations are over. So we invite all the participants to write down their questions and or comments in the chat feature of the Zoom. And we will address all these questions and comments at the end of the discussion session later on. So on that note, we will begin this webinar session. So our first speaker is Dr. Sylvia Gatsit. Welcome, Sylvia. Sylvia is now a senior scientist and team leader of the Liver Brain Unit, Rita Moretti, in the Fondazione Liver, sorry, Italian Liver Foundation in Trieste, Italy. I will do better next time. She obtained her PhD in molecular medicine in Trieste in 2008. Her work is on carnivorous spectrum disorder, focusing on identifications of biomolecular determinants of the disorder to improve diagnosis and therapeutic approaches. Since 2014, she's also been working on Parkinson's disease, looking at triggers of dopaminergic neuron loss, which is associated with movement, and potential pharmacological compounds to protect this loss. So I will now turn the session over to Sylvia to begin.
Thank you very much, Corey. I will share my presentation. Just a second, share. Okay. In presentation mode. Switching the videos, now I'm ready. As a first, let me thank sincerely for this invitation. It's a pleasure to share with us our most recent results, the work that we do in FIF. With the main goal today of my presentation, explain how FIF deals with the clinical needs of baby with severe neonatal hyperbilirubinemia and our ever needs in order to ask your help for continuing our research. Before starting with the data, let me show you the faces of the people that contributed and produced all the data I will show you today. So, as already introduced, today I will talk about bilirubin entry into and damage to the neonatal brain, leading to motor, cognitive, and auditory symptoms with a huge variability of severity. Despite carnicterus spectrum disorder, neonatal severe hyperbilirubinemia has been quite considered a no problem, a solved problem. In my opinion, this is not true, because if we calculate the number of baby deaths not suffering sequelae of neonatal hyperbilirubinemia, but death due to severe neonatal hyperbilirubinemia every year in the world, this number is incredibly high. It's quite 2 million babies. So the major reason why carnivorous is still present, as example, in the Western Europe, North America, is mostly due to the late diagnosis. It happens in the rural areas, it happens for cultural reasons. But now there is also a re-emerging carnivorous in preterm infants, possibly due to two major reasons. As a first, phototherapy is inefficient in preterm. Second, some treatment used in preterm care can displace bilirubin from albumin in blood, so increasing the risk of developing carnivorous. While in low middle income country, the lack and late diagnosis and the inefficient therapy are the major cases. So in my opinion, the translational needs possibly are not so different. We need improving the diagnosis and new therapeutic approaches. The first thing I believe we need is a more unified nomenclature. In the past, the effect of bilirubin on the brain has been defined by a lot of different terms: acute bilirubin encephalopathy, chronic bilirubin encephalopathy, carnivorous, bilirubin-induced neurological dysfunction, etc. We support the idea of Dr. Stephen Shapiro, Dr. Watchcombe in 2017, decided to define the plethora of symptoms due to bilirubin toxicity to brain by two definitions: acute bilirubin encephalopathy, or ABE, and carnicterus spectrum disorder, KSD, definition based on the pathophysiological and clinical features of the conditions. With acute bilirubin encephalopathy, we identify the symptoms in the first days up to 1 month of life of the baby, with very specific motor signs, opisthotonus, retrocollis, the paresis of the upward gaze, that may be reversible or proceed to Kernicterus spectrum disorder with a plethora of different level of motor, auditory, and cognitive defects that are permanent. This immediately suggests that bilirubin can affect different areas of the brain. But if we go deeper in the differences in between acute bilirubin encephalopathy and Kernicterus spectrum disorder manifestation, in between preterm and full term, we can immediately observe that they differ. In preterm, acute bilirubin encephalopathy signs are really subtle, making the diagnosis hard, while in full-term baby, we have the classical sign. KSD in preterm is predominantly auditory, while in full-term, the percentage of hearing loss is reduced, and we have mainly motor signs. This is a clue that the developmental stage of the brain at the time of bilirubin challenge is relevant to define what part of the brain and so what symptoms, what disability the baby can develop. Let me go through this table that I modified from a review of Dr. Jayanti to tell you one more very important translational point. As you can see here, phototherapy is fully able to reduce total serum bilirubin and free bilirubin in full-term infants. But phototherapy is absolutely unable to modify free bilirubin in preterms. As a first, let me explain better what free bilirubin is. When you perform a blood test for bilirubin, what you check is the total serum bilirubin. That is the sum of conjugated bilirubin that cannot enter a damaged brain and unconjugated bilirubin. Unconjugated bilirubin in blood is bound to serum albumin, a very big molecule, avoiding bilirubin entering the brain. So the minimal fraction of unconjugated bilirubin unbound to serum albumin is named free bilirubin, and this is the fraction of unconjugated bilirubin that can enter and damage the brain. The problem is that still we based our diagnosis on total serum bilirubin because there are no routine way to quantify free bilirubin in clinic as well as in animal models. There are ongoing some approval, FDA approval for free bilirubin point of cares. And in my knowledge, the only country that is quite routinely quantify free bilirubin in clinic is Japan. But nevertheless, without free bilirubin, we don't have a real direct estimator of the risk the baby is having. And total serum bilirubin is a suboptimal measure of the risk because low amount of albumin and everything can displace bilirubin from albumin increase the risk without the possibility for us to understand that this is ongoing. What FIF do for studying the regional sensitivity? There is no doubt that the amount of the challenge and the duration of the challenge are the key for determining the risk of developing permanent bilirubin-induced sequelae in infants. And this is the reason why an early and safe diagnosis and efficient treatments are fundamental. But let me debug a myth. It is not true that when bilirubin enters the brain, it will accumulate in specific region, the one that then will be damaged and lead to symptoms. The amount of bilirubin is the same among damaged and resistant region, both in the clinic and in models. How we can be sure of that? And it is true that the different regions have a different sensitivity to bilirubin that can maybe explain the variability of the symptoms. Yes, and as you can see in this experiment, when we prepared slices of different areas of a rodent brain, hippocampus, controlling learning, memory, and cognition, cerebellum, movement, inferior collicula on the auditory pathway, superior collicula, visual pathway, and is considered a resistant region, so a control, and the cerebral cortex, again, motor function, and we challenge the slices with a very specific amount of bilirubin, in that case, free bilirubin, because in the lab we can quantify free bilirubin, 70 nanomolar. At 70 nanomolar, only the hippocampus show damage. And we have to increase to 140 nanomolar, a frankly toxic amount of bilirubin, to see some reaction in the inferior collicula and in frontal motor cortex. Why the superior collicula, as expected because it's a resistant region, needs an extremely high amount of bilirubin. What that means? That means that there is no one threshold of sensitivity, but there are several thresholds of sensitivity, region dependent. If this is true also in the clinic, honestly, I don't know, because I don't have in my mind some study that try to correlate different amount of bilirubin, toxic bilirubin, to the symptoms. But might be possible that there is a risk of auditory dysfunction, a risk of cognitive dysfunction, a risk for motor dysfunction. One additional point of this study is that the cerebellum is never damaged after 24 hours of bilirubin challenge. And for producing some reaction from the cerebellum, we need to challenge the slices at least for 3-4 days. And this fully agree with the development of the cerebellar hypoplasia that is the hallmark in bilirubin toxicity in rodents, but rodents undergo bilirubin challenge for 8, 10, 15 days before showing cerebral hypoplasia. What that means? It means that the cerebellum is a region that needs a chronic exposition to toxic bilirubin to be damaged. And this can explain why rodents that we don't submit to phototherapy, cerebellar hypoplasia is present, but is not one of the major regions of the brain damage in the baby that hopefully will be soon treated. We have additional proof that the regions of the brain differs in their ability to respond to bilirubin? Yes. Still in fifth, we perform this extreme experiment by injecting the gun rats with sulfadimethoxine that shift all the bilirubin from the blood to the brain, as you can see here. And now, if you check the other colored curve representing how each region deals in cleaning, in oxidized bilirubin, it is absolutely clear that each region has a different dynamic. And this dynamic of bilirubin oxidation fully agree with the upregulation of cytochrome monooxygenase 1A1, 1A2, 2A3 that are known to oxidize bilirubin. And additional proof that later on after me, Dr. Gianpaolo Lido will describe better. A simple histology of different areas of the brain of the GAN rat, the model of hyperbilirubinemia. There are regions with specific manifestation of bilirubin toxicity. As example, in the parietal motor cortex, it is evident a decrease in the extracellular matrix formation. Necrosis happens only in specific areas of the frontal cortex and inferior collicula, while other mechanisms as a general cellular undifferentiation and a delay in brain development are shared among all the regions. How we try to understand if the developmental status of the brain can modify the toxicity of bilirubin. Coming back to the slices. Here we sliced the region of interest from a 2-day-old animal and exposed to toxic bilirubin. Here we did the same with slices from an 8-day-old animals. And as it is easy to see from the y-axis, the damage induced by bilirubin in 8-day-old animals is 4 to 5 times higher. So it means that, yes, the developmental stage at the time of challenges can modify the damage. Why that? Because the brain is not a one organ. The brain is a puzzle of regions, each one developing with a different time scale. As example, here, if you look at these gray squares representing the maximum activity of mechanisms key in brain development, you can see from the left to the right that the hippocampus is the first region developing after the birth, then the edge cortex, then the frontal cortex, and lately inferior collicula. Again, Dr. Gianpaolo Lido will enter into the details how bilirubin can affect the region during the development of the ganglions. But up to now, I discussed how bilirubin can modify gene expression. What if the opposite is the reason of variability? This take in discussion the third hypothesis for explaining the sensitivity to bilirubin. This hypothesis suggests that the individual genetic variability can be responsible for a different individual sensitivity to bilirubin damage of ability to resist to bilirubin toxicity. And thanks to our colony that actually is having hyperbilirubinemic animals, both with a very weak motor phenotype, and we named them low phenotype, LP, an animal with a frank phenotype, SP, severe phenotype. Again, Dr. Gianpaolo Lido will show you the details later on. We try to address also this issue because this phenotype correlate, as you can see in this picture, showing simply the dimension of the cerebellum, correlate with an increased brain damage, and most importantly, is heritable in our colony. So what we did is performing a genomic sequencing, comparing the severe phenotype animals with the weak phenotype animals and searching for differences. And we found almost 5,600 mutations specific to severe phenotype animals that after several steps of selection allow us to identify almost 800 mutations in 350 genes with a potential biological relevance. Why? Because among them we have stop gain, stop loss, and insertion deletion of up to 100 base pair, in addition to non-synonymous mutation that are the larger part of. So in order to gain some biological evidence of the potential effects of those genomic mutation, now we are moving to RNA sequencing, ever comparing the same animal, SP versus LV, and after the similar steps of selection, we hope to find 20 candidates to follow in the GAN rat, to be further reduced to 3-5 good candidates to validate in human sample. And the final goal is a diagnostic chip able to identify baby that for their genetic background are extremely at risk of carnitine spectrum disorder, even at bilirubin level, possibly below the risk threshold. And for that, we are asking for your collaboration, if you are available, because despite we have clinical partners, we ever need more clinical partners, especially if we are discussing about individual genetic variability, where samples from different areas of the world can be absolutely important to identify the real key genes involved in the genetic susceptibility. But up to now, I deal only with the differences among the region. What is perfect for biomolecular identification of the determinants of bilirubin damaged brains, to better understand the disease and possibly to improve the diagnosis through the gene. But maybe this approach is not optimal for a new therapeutic option. The best way in our mind for finding a new therapeutic option is searching for the general mechanism, damaging pathologic mechanism, shared by all the regions. And we did that both in the slices, identifying redox stress, inflammation, and glutamate neurotoxicity as the shared general mechanism responsible for damage in all the regions we addressed. And we repeated these results in vivo. So after knowing there are targets we can try to reverse bad drugs, we tested as a first in vivo if single antioxidant, single molecules able to block glutamate channel and anti-inflammatory molecules can reverse the damage. Here you have to compare this line, this yellow line that is the damage observed in slices treated only with bilirubin, with the colored one bars representing the single treatment. Despite there is an improvement, we are far from this black control line, the healthy slices. But we approached the control slices when we combine the three drugs in a cocktail. This means that bilirubin toxicity plays simultaneously across these three mechanisms, and that if we want to fully protect the brain, we need a cocktail. We need to fight to reverse these three mechanisms simultaneously. And we repeated this scheme, this experimental scheme in vivo in the GAN rat. So now you have to check this black line that are the normal bilirubinemic control, this yellow line that is the cerebellar hypoplasia in hyperbilirubinemic animals not treated. We treated again single drugs against glutamate neurotoxicity, no effect. Inflammatory drugs, ibuprofen, no effect. Indomethacin, even worsen, significantly worsen cerebellar hypoplasia. And acetylcysteine, chosen as an antioxidant, was not well tolerated by the animals, so we moved to a second nutraceutic, curcumin, that we selected as an antioxidant. As you can see here, by looking at this red bar, we fully protect the cerebellum. And as additional proof, we perform behavioral tests that demonstrate that, again, looking at the red bar compared to yellow hyperbilirubinemic animals not treated and black controls, curcumin fully restored the behavioral capability of the animals, suggesting that the whole brain was fully protected. But how curcumin, that we choose an antioxidant molecule, can do that? Again, compare the red bars with the black controls and yellow untreated gum rats. Curcumin protects by a pleiotropic, anti-inflammatory, anti-redox, anti-glutamate action, and is able to normalize even more markers of bilirubin damage that we follow without modifying the amount of total serum bilirubin. What that means? It means that we have really a good candidate in clinical perspective to use in babies to avoid the brain bilirubin damage, even if the baby is still under toxic total serum bilirubin, still under a toxic challenge due to bilirubin. And curcumin is really interesting in a clinical perspective because it's a nutraceutical that has been largely used in clinic, also in infants, where it shows no significant side effects. Even more, it's lipophilic, so can be easily diluted in the milk in the baby bottle. And again, we are happy if someone of you won't join us in this project. So, as a final resume of my presentation, how FIF deals with the three major hypotheses explaining the variability and the individual sensitivity of babies to bilirubin neurologic damage. About the regional sensitivity, yes, we demonstrated that it's true, and we suggested that maybe there are different bilirubin, total serum bilirubin threshold, explaining the prevalence of motor, cognitive, of auditory symptoms. And most importantly, we identify common pathologic mechanisms that we can reverse by curcumin. About the developmental sensitivity, I let you follow the presentation of Dr. John Paul Lido after me, but again, the first trial we did curcumin showed that curcumin can really protect the brain, and a final proof is ongoing. About the genetic susceptibility, yes, we have some data based on our colony. In my opinion, actually, the stronger proof comes from our collaborators in the clinic, especially Prof. Shapiro and Dr. Jordan, but it's fully believable that the individual genetic variability can make some baby more prone to damage or the baby fully resistant. And we are deeply working in identify those candidates for finally having a diagnostic chip that can improve the diagnosis. So finally, let me thanks all our collaborators and supporters, and most importantly, all of you for your attention. Thank you very much.
Okay. Thank you, Sylvia, for the excellent presentation and in keeping on time within 30 minutes. So we just learned a bit more about the Kernicterus spectrum disorder particularly on the common pathological mechanisms of bilirubin toxicity in the neonate's brain, and the particular need to develop a diagnostic kit that can enable us to identify babies with potential disorder, even though they have possessed low bilirubin levels on the blood. So before we move on to the second presenter, we would like to remind the participants to please write down their comments or questions in the chat feature of the Zoom meeting, and also in the comment section in the YouTube live webinar. So with that, let's go to introduce our second speaker, which is Dr. John Paul Guido, who is now currently a PhD candidate in University of Trieste and the Liver Brain Unit Rita Moretti in Fondazione Italiana Fegato. John Paul obtained her Bachelor of Science in Biology and also Master of Science in Molecular Medicines in Manila, Philippines. He has previously been working with different in vitro models to study dengue and influenza viruses, and also in vivo model to study immune response against hepatitis C viruses. Since starting his PhD in 2021, he has now published 2 first author papers in international peer-reviewed journals. I will now turn the session over to John Paul to begin.
Hello, good morning, everyone. Thank you, Cory, for a very kind introduction. Let me share my screen. All right. Then, okay. Again, good morning, everyone. For today, I'll be presenting our work on bilirubin-induced transcriptomic imprinting in neonatal hyperbilirubinemia. So from the previous talk, definitely severe hyperbilirubinemia is resulting to bilirubin-induced neurotoxicity to several mechanisms. However, none of these mechanisms have fully explained the variability of symptoms that observe in the clinics. Here in fifth, we are using the GAN rats as our animal model for hyperbilirubinemia and bilirubin-induced neurological damage to try to answer the question. So the GAN rats exhibit UGT1A1 mutation, which results to the lack of the activity of the UGT1A1 enzyme that results to hyperbilirubinemia, which causes bilirubin-induced damage in the brain. And the hallmark of the damage in the GAN rats is cerebellar hypoplasia. So, however, cerebellar hypoplasia is usually absent in the clinic, where hyperbilirubinemic neonates develop symptoms of auditory, motor, and cognitive dysfunction. For this reason, we have moved to study the different regions of the brain. Example here, the frontal cortex, which contributes to decision-making and motor movement. Parietal cortex also contributes to movements, hippocampus for learning and memory, and social, spatial, and learned movement. And finally, the inferior colliculi with auditory functions. In our study, we want to understand how bilirubin impacts the regional and the development of brain that correlates with the affected functions in Kernicterus spectrum disorder. As you can see here in the slide, frontal cortex and inferior colliculi apparently share common features. Zoom in here. For example, the white spots that you're seeing here in the histological slide at P9 suggest necrosis, while the darkly stained cells suggest the appearance of microgliosis. And also, finally, when we counted the cells, there's an increase in the cell number, in the overall cell number in the hyperbilirubinemic animals and P9. In addition, we observed that in the frontal cortex and the inferior colliculi, they also share the apparent recovery of the structures in the adult age. Moving on to the parietal cortex. Moving on into parietal cortex at 9 days after birth, we have also observed that at P9, in hyperbilirubinemia animals, there's a decreased layer of cell thickness. Here, it's obvious, and increased cell density. And here in the arrows, the red arrows indicating the defined layers, that's also no layer definition in hyperbilirubinemic animals, and the lack of heterogeneity, which suggests that there's a delay in the extracellular matrix formation and cell differentiation. And similar to the first two regions that we have discussed, there's an apparent recovery of the structures in the adult age. The next and the last region that we included in the study is the hippocampus, where there's also a significant difference in at P9 in normobilirubinemic and hyperbilirubinemic animals in terms of cell shape, cell organization, and lack of their heterogeneity. And similarly to the first three regions that we have been shown, there's also an apparent recovery into adulthood. So, and again, this suggests a possible brain developmental delay in hyperbilirubinemic animals, and both are also apparently recovering in adults. In adults, yes, yeah. So to summarize everything, that all the featured characteristics, here you can see that necrosis, microgliosis, and increased cell number are shared between the regions of frontal cortex and the inferior colliculi, and the hippocampus and the parietal cortex, the H cortex, share the lack of heterogeneity and delay in cell differentiation. And there's a markedly region-specific feature in the H cortex comprised of these four, which suggests delay in ECM formation, while all the regions share an apparent recovery into adulthood. So at first, this seems quite new, but we are not surprised since this has been previously demonstrated that half or almost half of the genetically modulated genes that control brain development in the cerebellum of hyperbilirubinemic animals. So Vianello has already demonstrated this, and from her research, we have selected 20 genes that play a role in different brain developmental processes, such as synaptogenesis, synapse formation, migration, differentiation, repair and plasticity, and ECM formation, and some genes that plays a role in the behavior. And here we can show the region-specific mRNA transcription levels during the postnatal development of hyperbilirubinemic gun rats versus their age-matched controls. So here, apparently, with this data, apparently there's seemingly no rule that is that we can observe, except that all of the genes are modulated in all postnatal ages. To give more meaning and to better interpret the data that we have, we would, thanks to Dr. Davis Pascut, we perform correlational analysis where the clusters of genes that show the same trends or patterns here to study, to show the same trends and or expression patterns. So for example, here you can see the cluster of the genes of HIL4, NTSR1, BMP5, and calmodulin in NJ is apparently disrupted, and they are dispersed in terms of their correlations, in terms of correlational hierarchy. So since we're studying the brain development, this suggests that this perturbation suggests that there's an abnormal postnatal development in hyperbilirubinemic animals. In addition to these observations, here you can see the yellow circles as pointed by the arrows, which signifies negative correlations between genes. So to give you an idea, we say negative correlations in between the two genes. For example, across the postnatal ages, they have a different pattern. For example, one gene is upregulating while the other gene is downregulating. While for the positive correlations, both genes are seem to have the same pattern of upregulation across the postnatal ages. So here you can see that the yellow circles in arrows is absent in hyperbilirubinemic animals, which again suggesting the inversion of the correlations that are found in normal bilirubin animals. And based on this data, and we found that this is pretty much observed in all the regions of the all the brain regions that we have studied. In addition, we found that most genes are only transiently modulated in the inferior colliculi, in the hippocampus, in the frontal cortex, which suggests that they are potentially correlate with the acute bilirubin encephalopathy. While in the parietal motor cortex, we found that 16 out of 18 genes that we have studied are permanently altered in the adults, which suggests or in agreement with the connectorosis spectrum disorder. To further provide the biological evidence of these transcriptomic modulations, we also perform behavioral tests. Here you can see that hyperbilirubinemic animals in orange color show significant motor deficits in different postnatal ages of development until adult age, supporting the transcriptomic data or results that we have provided. So this gives us, this suggests that there's something going on during the brain development and that significantly disrupted by bilirubin concerning motor control. Here in this slide, to summarize, earlier we have shown you the histological slide of the parietal cortex. Especially at P9, we see a significant reduction of the layer in the JJ. And here, the apparent recovery in the adulthood at the histological level is in contrast with the results that we have found at the transcriptomic level and the behavioral results. So this data, the transcriptomic data and behavioral data that we found, apparently is suggesting the evidence that the histology that we observe in the adult, apparent recovery in adult, is not true. So to summarize the data that all the results, the data that we found, we found that each region responds differently to bilirubin. And the effects of bilirubin in the frontal cortex, hippocampus, and the inferior colliculi are apparently transitory in effect, which should correlate with the acute bilirubin encephalopathy, while the effect of bilirubin in the parietal cortex is shown to be permanent. But overall, there is the perturbation, the presence of perturbations in postnatal brain development in hyperbilirubinemic animals. One more important finding from our work is that we found that GRM1, a gene responsible for the expression of a glutamate receptor, is the only permanent downregulated genes in all the regions on the study. And we know that glutamate receptors are crucial in neurotransmission and brain functions. Despite knowing or identifying many genes that are significantly modulated, we still would want to understand and have a suitable candidate to explain the variability that we observe in the phenotype. And fortunately, we have here at FIF a useful and powerful tool in the form of our grand rat colony, especially the hyperbilirubinemic animals that develop a distinct severe motor dysfunction compared to...
Cuma mati doang.
Sorry. Okay, so with a distinct severe motor dysfunction in the SP that is that can be shown here.
Yang itu udah pasti mau kan.
Okay, so here, to give you an idea of what we're talking about, these are both hyperbilirubinemic animals, but you can see here the performance of the LP and the beam walking test. So the beam walking test is for the animals to cross the beam, and we take a video and measure how fast and how well they do the test. Let's see again the... I'm not sure if you're seeing it, if it's playing. But here, for those of the audience who maybe the internet is lagging, the LP animals crosses the beam, the wooden beam, very fast compared to the SP, where the SP animals are struggling with crossing the beam, and sometimes their paws are falling off the beam. So what's the significance of this observed phenotype? So let's go back for a bit in showing the different symptoms observed in the clinics. So these are some of the variable symptoms that is observed in the clinic due to Kernicterus spectrum disorder, thus giving us an idea that these animals are really good model to find to further study the variability of the symptoms that we would like to elucidate. So when we quantify the beam walking test, here we've shown the speed of the performance, and we found that there's a significant worsening of the condition in the SP animals, which is also shown or evidence with when we check the cerebellum weights and size. Here you can see in the photo, there's a really obvious and evident reduction of cerebellum size that is also reflected and significantly different in terms of cerebellum weights. So now we decided to perform again gene expression analysis in these animals to try to understand which among the significantly altered genes are really responsible for the worsening of the condition. And here we included cerebellum, since it's a gold standard for the gun rats, since it's a hallmark, cerebellar hypoplasia is a hallmark in hyperbilirubinemic gun rats, while for the parietal motor cortex, we found earlier, as we have discussed, that 16 out of genes are significantly downregulated in this region. Here, we show this is our preliminary data of our gene expression analysis of in the cerebellum and the parietal cortex region. And for easier view, we summarize here that our findings. First, 8 8 out of 20 genes we found that are significantly different in SP. So these 8 genes are signified by the asterisk here, which give us our 8 candidates that could explain the worsening of the condition. Secondly, here when you look at the arrows, there's shows a region-specific pattern where in the cerebellum there's mainly upregulation of genes, and while in the H cortex there's mainly a downregulation of genes. And third, the most important and interesting finding that we observe is that GRM1 is again the only gene in both regions that is downregulated in LP and SP animals. Okay, so we will study all the genes, all these candidate genes, to really confirm if they play a role and if they could explain the appearance or the severe phenotype that we are observing. But for now, we can share with you our preliminary proteomic data for cerebellum. As you can see here in the cerebellum, the amount of GRM protein is evidently decreased in severe phenotype here in red. And even here you can even cannot even see one of the bands. But overall, there's an evident decrease in this protein, confirming the potential dysfunction of glutamatergic synapses in the severe phenotype animals. So we are still doing this work is still ongoing, but aside from proteomic analysis, we are also doing or performing now immunofluorescence microscopy, where the goal is to see the distribution of the protein targets, for example, the GRM1 and the MAP2, to assess the synapse and neuron morphology. So for us to better understand the more clear picture of what's really happening at this level. In addition, we also plan to count the number of cells and neurons that could also give us important information on the overall picture of what's really happening in the severe phenotype animals and what could contribute to the variability of the symptoms. Of course, we are trying to find the explanation on why we observe this phenotype. And we know that severe hyperbilirubinemia is a strong risk factor in developing Kernicterus spectrum disorder. So in this regard, we also measure the total serum bilirubin in the rats, and we found that between LP and SP, there is no significant difference between the TSB levels of these animals. But we know that for the damage to occur, the bilirubin must enter the brain. So we also these are preliminary data, and we are waiting for additional results. But here our preliminary data shows that there's also no significant difference between LP and brain bilirubin in the LP and SP animals in cerebellum and parietal cortex. And since we have the opportunity to also measure the brain bilirubin in other regions, we found the same thing. So the TSB and the brain bilirubin concentration could not fully explain the observed severe phenotype that we have. So the work is still ongoing, but of course we continue to look forward in other ways to explain variability to explain the Kernet spectrum disorder. At the moment, as Sylvia have already discussed, we have sent RNA sequences. We are analyzing RNA sequences as well to hopefully find a severe phenotype specific variants that hopefully when we have this number of candidates can also be translated to the clinics and check and validate. Yeah. So that ends my presentation. But before anything else, I would also take this opportunity to acknowledge and give thanks to all the people that made this study happen. From, of course, the Fondazione Italiana Fegato, specifically here, I'm at the liver brain unit where we are conducting this work. Our university, where I'm doing my PhD in molecular biomedicine, our collaborators in Prague and in France, and definitely my funding agency in the Philippines, because I will not be able to be to get this opportunity without this very generous funding that they give. And also to all the people that not only makes do incredible science, but also makes things bearable while we are dealing with a lot of experiments and trying to analyze the data that we have. So and with that, and of course, for BRIN also for inviting me to share our work, and also special thanks to Shri, my mentor as well. She taught me some experimental techniques in the lab when I first arrived here. So thank you, Shri. And that's all. Thank you so much for your kind attention. And yeah, that's it. Thank you.
Okay, sorry. Thank you, John Paul, for the presentation, and apologies for some connection issues earlier. So we have just heard the presentation from John Paul regarding his study on transcriptomic analysis on gun rat models with different phenotypes of hyperbilirubinemia with either low or severe motor dysfunctions, and identifying possible genes that are differently regulated in these conditions. So we again invite all the participants to type their questions or comments for each of the presenters, and we will address these comments and questions in the discussion session later on. So we're moving on to our last speaker, last but not least, our own Dr. Sri Jayanti, PhD, who is currently the postdoctoral fellow in the Eichmann Research Center for Molecular Biology, working with viral-related liver disease and other viral infectious diseases in our research group. Sri obtained her MD in 2015 from University of Hasanuddin and her PhD from University of Trieste and the liver brain unit Rita Moretti in the Fondazione Italiana Fegato in 2022. She now joined our research center since 2023. I will now turn the session over to Sri to begin.
Thank you, Mbak Kori. Thank you, Bu. And I will try to share my screen. Okay. Good afternoon, everyone, and good morning for you in Italy. And today I will continue to discuss the bilirubin studies. If Sylvia and also JP have previously discussed and explained to us about the bilirubin neurotoxicity, I will try to discuss another side of the coin. I will explain our project regarding bilirubin neuroprotective effect in neurological diseases, particularly Parkinson's disease. Okay. So a little bit introduction about bilirubin, the yellow pigment. So bilirubin is the end product of heme catabolic pathway, and it has two forms. First is the unconjugated bilirubin form, which is the form that is not water soluble, and therefore it needs to be transported to the liver to make it water soluble, and what we call as conjugated bilirubin. But today we will focus our discussion for this unconjugated bilirubin. So actually this bilirubin has two phases, or what we call as genus phase. First, it's known as a toxic pigment, especially in neonates, because it's induced severe brain damage, as Sylvia and also JP have mentioned before. But in the last 2 decades, it has emerged that bilirubin also has neuroprotective effect, especially what we observe in Gilbert syndrome, where in this population, they have lower prevalence of chronic diseases. And what is actually Gilbert syndrome? So Gilbert syndrome is a condition that characterized with a mild elevation of unconjugated bilirubin. But in this population, they have no signs of hemolysis, or even they have no impairment of liver enzyme activities. So simply they just being jaundiced. But in this GS populations, this condition is primarily due to the mutation of UGT1A gene, the gene which is responsible for the conjugation of bilirubin in the liver. If general population usually has UCB levels around 0.2 to 1.2 milligram per deciliter, in GS subjects, the UCB level can reach up to 4 times higher of UCB level compared to general populations, and no treatment is either needed or recommended for Gilbert syndrome subjects. And then from this Gilbert syndrome, we learned that they have lower prevalence of chronic diseases, including they have the low prevalence of ischemic heart diseases compared to general populations. And also they have better insulin sensitivity, and also they have lower risk of metabolic syndrome, and also the type 2 diabetes. And also they are more leaner, compared to general population. And this Gilbert subject also found to have a lower risk of cancers. So that's raised the question, what we observe in this Gilbert subject then raise the question, what's the potential clinical implication of hyperbilirubinemia? And actually it has been demonstrated in a paramount studies that bilirubin has multiple biological functions, including it's known as a powerful antioxidant, and it also has anti-inflammatory and immunomodulatory effects, and also it plays a role in multiple cell signaling process, and nowadays even bilirubin is now considered as a hormone. Okay. And this knowledge about bilirubin clinical impact in chronic diseases has also bring an interest in another diseases or neurological diseases. Why? Because the clinical data have shown that reduced serum bilirubin level are present in various neurological disorders, suggesting that a loss of bilirubin clinical benefits will bring more harmful for people. For example, in Alzheimer's disease, dementia, and amyotrophic lateral sclerosis, multiple sclerosis, and cerebral infection, and Parkinson's disease, patients usually has lower serum bilirubin level compared to the healthy controls. And this condition then raises also another question, that food targeting bilirubin will provide protective benefits for patients with neurological diseases. And in FIF, we try to address these questions, and we choose to perform experiment regarding bilirubin protection in Parkinson's disease. And why we choose Parkinson's disease? Because Parkinson's disease is a brain condition that can causes problems with movement. Usually they have a motoric movement disorder. For example, they have tremor, they have slow movement, and they have walking gait or postural instability. And this disease usually occurs in older people, but younger people also can be affected, and mostly found in men. Then Parkinson's disease is a chronic progressive disease, and it gets worse over time. But till now, there is no cure other than symptomatic treatments. And it also results in high rates of disability and an increasing need for care, which will lead to a personal, social, and also economic burden for patients and also their families. As healthcare improves, elderly populations also increases, and so does Parkinson's disease prevalence. In 2019, there were over 8 million individuals with Parkinson's disease globally. In Southeast Asia itself, it has been reported that more than 500,000 people live with Parkinson's disease. And what is the challenges in Parkinson's disease? So first, the prevalence of the number of people live with Parkinson's disease is expected to be double, and it's even estimated to be a non-infectious pandemic in 2040. However, till now, we still rely to diagnose Parkinson's disease, we still rely on clinical symptoms because until now there is no specific biomarker available, and unfortunately there is no disease-modifying therapy for Parkinson's disease. And what happened actually inside the Parkinson's disease brain? In the midbrain, in the area of basal ganglia, there is a region called substantia nigra. The substantia nigra contains dopaminergic neuron, which produce dopamine. Dopamine is a neurotransmitter that's responsible for motoric movement. In Parkinson's disease, there is the reduction or the death of this dopaminergic neuron in this substantia nigra, and it appears in the clinical symptoms for patients that they have this motoric movement disorder. And multiple mechanisms have been proposed to be involved in the death of dopaminergic neuron, including oxidative stress, neuroinflammations, and also protein misfolding and aggregations. And then, knowing that UCB has an antioxidant, anti-inflammation, and also neurotropic effects, which probably can address Parkinson's disease pathologies, including redox stress and neuroinflammation. At that time, in fifth Italian Liver Foundations and under Liver Brain Unit of Rita Moretti, during my PhD, together with teams, Prof. Tiribelli, Dr. Gassin, and Dr. John Paul, and also Emanuela, we began to investigate the effect of UCB in Parkinson's disease. And to study this UCB effect on Parkinson's disease, of course, we need a reliable experimental model that can mimic Parkinson's disease. For this reason, we use a previously developed organotypic brain cultures, a previous ex vivo model that developed in our group. We use organotypic brain culture of substantia nigra from Wista rat. We treat it with low dose of rotenone. What is rotenone? Rotenone is a pesticide that's known for its ability to induce Parkinson's disease in humans. And using this ex vivo model, we can maintain the architectural heterogeneity of substantia nigra, and when it's also allow us to mimic the chronic progressive Parkinson's disease in humans. And it's marked by the reduction of dopaminergic neuron, as you can see here, along the time courses of treatment. And interestingly, this model also allows us to unravel multiple mechanisms involved in different time courses, including inflammations and also redox imbalance, especially in pre-diagnostic phase. It's an important time courses for us because by this way we can understand which pathomechanism that triggers the loss of the dopaminergic neuron. And then we treat unconjugated bilirubin or UCB to see its protection in this ex vivo Parkinson's disease model. To determine the protection, we count the dopaminergic neuron in this model. We perform the counting by using immunostaining techniques, and this is the graph of the number of dopaminergic neuron. As you can see here, blue bar indicates the MSO or treated slices or control, and the gray bar indicates slices treated with rotenone. And as you can see here, in the rotenone treatment, we found there is a reduction of dopaminergic neuron up to 40%, and it's mimicking the diagnosis stage of Parkinson's disease patients. And as we treated these slices with various range concentrations of bilirubin from 0.5 to 4 micromolar. And as you can see here, among all the concentration used, 0.5 and 1 micromolar UCB are protected dopaminergic neuron from rotenone-induced dopaminergic neuron loss. So in this stage, we found that UCB can protective in Parkinson's disease with these concentrations. And since we know that UCB apparently can protect dopaminergic neurons, of course, we need to understand in which way UCB protect the dopaminergic neuron loss. We tried to match this dopaminergic neuron count that we find previously with the mRNA expressions of oxidative stress, neurotrophic factor, and inflammation markers that known to alter in Parkinson's disease. And among all these markers, as we can see here, the one that only fitted with the protection that over by UCB, only the alteration of TNF-alpha. So here we conclude that probably UCB 0.5 and also 1 micromolar prevent the dopaminergic loss of neuron by acting as anti-TNF-alpha. But we have another question because we know that UCB is a powerful antioxidant. So why it cannot work as an antioxidant in this model? Or why it doesn't possess the antioxidant activity? And we try to answer this question by treating this rotenone ex vivo model of Parkinson's disease with another antioxidant treatment, which is an acetylcysteine. It's also a powerful antioxidant that used in the clinic. And as you can see here, we use a very wide range of concentration just to make sure that what is the role of antioxidant in this Parkinson's disease model. But as you can see here, none of NAC concentration was able to increase the survival of DOPA. And it means that actually the oxidative stress is not the key player in this dopaminergic neuron loss in this Parkinson's disease model. And then we tried, since we hypothesized that TNF-alpha probably the key player, and then we try to answer this hypothesis by using two approaches. First, we treat the BRIN slices, the healthy BRIN slices with TNF-alpha only in different concentrations. And as you can see here, by treating with TNF-alpha, by treating the cell with TNF-alpha, it can induce a concentration-dependent decrease of dopaminergic neuron number. And it also has similar dopaminergic loss with rotenone treatment. And as a counter-proof, we use the second approaches. We use the infliximab. Infliximab is a chimeric monoclonal antibody that acts to neutralize TNF-alpha. And when we co-treated this infliximab with rotenone, we can see that infliximab fully protected dopaminergic neuron from rotenone-induced death. So as a conclusion from this, our first part of experiment, we found that TNF-alpha plays a major role in dopaminergic neuron demise, and UCB protect dopaminergic neuron loss by acting as TNF-alpha in Parkinson's disease. But we have another issue to tackle. UCB can protect, but how to modulate bilirubin or UCB in Parkinson's disease? Actually, there is three ways that we can use. At first, we can use a systemic inducer to induce enzymatic machinery to produce bilirubin more in our body, but inducing MOX1 or inducing the production of bilirubin in systematic level, it can be done. However, the amount of UCB that can reach the brain is, we never know how much it can enter the brain. And the second approach, we can use a local modulation of bilirubin production in the brain, again by inducing heme oxygenase. But the problem is when we induce the production of bilirubin in the brain, we will produce not only bilirubin, but also we will produce iron, and it will enhance the damage in the brain. So the most feasible option that will be the most feasible option for us at the time is the using targeted delivery of UCB into substantia nigra by using a nanoparticle approach. And that's the second project that we performed in collaboration with University of Torino, who previously developed these nanobubbles that can care as a drug carriers for central nervous system. And with the help of University of Torino, we have the 3 formulation of nanobubbles. We have 3 different polymer cells of nanobubble. We have at the first one, we have glycol chitosan, which is the natural polysaccharides, and it's biodegradable. The second one, we have the GC with the addition of deferoxamine that can chelate iron. And we have the third one, which is the sophisticated one because it has a superparamagnetic iron oxide nanoparticles, and it can be driven in paramagnetic field, and it make this nanobubble easy to deliver and to pick up. Before we load the nanobubbles into this, before we load the UCB or bilirubin into these nanobubbles, of course, at first we need to understand if these nanobubbles are safe for our slices or the healthy slices. And to understand its safetiness, we perform the treatment of these 3 different nanobubbles with 3 different concentrations. Here we use the dilutions, different range of dilutions, and we compare to the MSOS positive control and rotenone treatment as negative control. And as you can see here, we can understand which nanobubble is actually the better one because GC is toxic to dopaminergic at 1 to 8, and GC-DFO is safe at least at 1 to 192 dilutions. Meanwhile, unfortunately, GC-DFO spions we need to exclude because during the experiment we found that it has a agglomeration event. After we found the most protective nanobubbles candidate, and then we continue to load UCB into these nanobubbles formulations. And again, we treated the ex vivo Parkinson disease model with these nanobubbles UCB formulations with a different range concentration. We also increased the dilution of these nanobubbles to mimic the low concentrations of bilirubin. And as you can see here, GC-DFO UCB at this concentration, 1 to 4,000 something, dilution gave the highest protections among all the formulation and concentration of nanobubbles UCB that we have. And though it's not a full protection because we only reach 80% something of dopaminergic neuron.
I think it's frozen.
Yeah, sorry, Prof. I'm disconnected before. I will try to share again my screen. Which is the last screen that we have? This one, I guess. This one? Okay, so nevertheless, even though we don't reach the full protections in this experiment, however, this experiment allow us to narrow down which the most protective nanobubbles UCB in this Parkinson disease model. And it's also given us the possibility to continue the experiment because apparently it's feasible to deliver UCB by using nanobubbles. And as a conclusion, our experiments give us an opportunity to investigate and to find the result that UCB at low concentration prevent the dopaminergic loss in ex vivo Parkinson disease model, especially working as an anti-TNF alpha. Yes, indeed, and there is a challenge on how we can deliver bilirubin in Parkinson disease, and we try to answer this issue by using nanobubbles approach as UCB carrier in Parkinson disease. And it seems a promising delivery system, and we found the safest and the most protective candidates. But of course, further studies are needed to understand more about these protective effects of nanobubble UCB. And eventually, of course, in vivo studies are needed to complete the story of this UCB protection in Parkinson disease. And okay, then let me acknowledge all the individuals and institutions that involved in this research, especially for Italian Liver Foundation, all the members, and also especially the Liver Brain Unit, Rita Moretti, Prof. Tiribelli, Sylvia, and also John Paul. A brief story that this Rita Moretti name is named after the late Dr. Rita Moretti, our beloved and very helpful collaborators in our research. And I also thank University of Trieste, Animal Facility of UNIS, University of Turin, University of Hasanuddin, also LPDP for funding this research. And last but not least, I also thank BRIN and also Ekman Research Center for molecular biology, because now I'm working here and I'm really happy because you give me a chance to introduce this study at these occasions, and perhaps and hopefully we can do more collaborations. We can do more collaborations and we can expand the research itself. Thank you so much.
Thank you, Sri, for the excellent presentations, and we apologize again for the connection issues earlier. So with 3 presentations on the potential use of bilirubin as therapeutic agents in Parkinson's disease, we are now arrived at our discussion session for today's webinar. So we have today at least 2 questions in the chat for Dr. Gadzin. So Sylvia, have you read the questions, or do you want me to read it to you?
No, yes, Corey. If I remember correctly, the first question is about the curcumin formulation we used and the bioavailability of curcumin. So this is the point we are working now, because in the first paper showing the food protection, we used a commercially available liposomic formulation of curcuminoids. And this is know that liposome increase bioavailability. So now we are trying again with alcoholic extract of curcuminoid, no formulations, with the hope that the literature that we know is true, and literatures performing rodents suggest that most important than the formulation is the posology. I mean, if you injecting the animal, administrate curcumin to babies in small quantity, but in a chronic way, the accumulation in the brain is good, while acute posology is completely inefficient because immediately cleans from the body. And what we did in the first paper was quite a chronic administration, because we injected the animal from P2 at the appearance of hyperbilirubinemia, mimicking a baby in a hospital, up to 17 when the cerebral hyperplasia has to be present if curcumin doesn't work. Now we are shortening the injection from P6 to P10 and using curcuminoid extract. Let's see what happens. So if I remember correctly, second question is a little bit more complicated for me because please remember I'm a biologist, not a medical doctor. And the second question is about
Go ahead, Sylvia, it's fine.
Bacteria. I will ask for your help in the case, okay? Okay, second question is about bacterial infection, curcumin and bilirubin. So first part of the question, I don't know exactly if all bacteria infection can increase bilirubin, total serum bilirubin. I can suppose it depends from the location inflammation. If the liver, probably yes. But bacteria induce inflammation, possibly even sepsis. And for sure, curcumin is an anti-inflammatory and with proven efficacy in models that sure also in sepsis and in clinic for different conditions. Even more, if it is inflammation, especially sepsis, this is the potential of opening the blood-brain barriers. And in that situation, even normal amount of bilirubin can enter, adding neurotoxicity to the inflammation due to sepsis. So in my opinion, we need clinical trial with curcumin. There are clinical trials, but in my opinion, few, and so possibly people need to be more convinced about the efficacy and especially the safety of curcumin also in neonatal care.
Thank you, Sylvia. Dr. Santi, would you like to comment on your questions?
Thank you.
Okay, no further comments from Dr. Santi. Okay.
Sorry, Corey was a little bit absent.
It's fine. Okay, thank you, Sylvia, for the excellent response for the question. So we move on to the next questions for Dr. Lido. From Cecilia, is your transcriptomic data from the animal equivalent to human? And is there any data available in the literature? John Paul?
Yes.
Yeah.
Okay, thank you for the question. So again, I will read. The question is if my transcriptomic data from the animal is equivalent to human, or is there any available data in literature? As far as I know, let's say at the transcriptomic level, there's no, sorry. Okay. There's no equivalent data yet, but at the clinical level, for example, the GRM1, which is glutamate receptor, which is also being detected in MRI imaging, our data is kind of giving or confirming or in alignment with the observed increase in signal that is observed in the MRI imaging when they try to detect glutamate. Okay, and most of the data that we have is not really like the focus is not really yet on the Kernicterus spectrum disorder, but the focus is more on the schizophrenia and ADHD. But in our study, that's why our study, especially the third hypothesis, is really important, because since we don't have any database yet or available database yet, any findings from the sequences that hopefully will align to our current findings at the transcriptomic level will enable us to have some good candidates that we can also check at the clinical level. So that's why we're really excited with the results of our work, because since incidentally, we found that GRM1, that is, which expressing the glutamate receptors, is also, you know, the one that is also found in at the clinic. So that's why we're excited to continue the work and perform the RNA sequences, and hopefully we'll manage to have and check if indeed we can have the equivalent data that we can check from the variants that we see, if it's hopefully in the future translatable to humans. But ultimately, that's the goal of our project. Thank you.
Okay, thank you, JP, for the explanation. Is there anyone who would like to comment or question directly from the audience? Please raise your hands or just open mic.
Can I ask?
Go ahead.
Okay, thank you. I'm really curious about JP and also John Paul and Dr. John Paul and also Dr. Sylvia Gatsen. As they discussed before, that there is one gene that altered constantly or permanently in this GAN rat population, right? And it's probably related to Carnot spectrum disorder. Do you have any plan in the near future for exploring this alteration in the clinical samples?
Yes, definitely. And I think we're really have high hopes that with this webinar, we can offer new projects to collaborate with, because at the moment, since we're working at the animal models, but the ultimate goal is to also translate this into the clinic. But of course, we need the samples and also clinicians that are really would like to take part and participate. Yeah, like Shri.
Yes, I will be very glad to involve. But which sample that you used before? Is it from blood, or the alteration you can find both in blood or blood and brain samples, or?
Okay, since we're using the animals models for now, we collected brain tissue, and we selected some the most interesting regions. But hopefully that's the next step. Once we have the variants, the list probably it will be like hundreds or maybe thousands, but hopefully we'll manage to select a few that is really specific to the severe phenotype, and hopefully those candidates we can check initially, like prioritize them in checking at the clinical level, maybe using blood or any available sample. But that's the plan. But at the moment, yes, we collected brain tissue for our study.
If I can add something to JP, we really hope to be lucky, because the genomic data arise from tail, whatever, because definitively it's genomic DNA. And this is important because thinking to a baby, to a translational approach, ideally, we can simply check the individual genetic variability through the blood. We hope definitively some of the variants may agree with the genes, the gene that John Paul is studying in the brain. If you remember the last slide presentation of John Paul, nor bilirubin in the blood, nor bilirubin in the brain explain the variability. So maybe some mutation occurred in one of the genes that is targeted in the brain by bilirubin, making this gene more damageable. So if it is true, we have in one shot both the genomic mutation to follow using blood and biological information of the role of this mutation in brain-induced bilirubin toxicity. So let's see what happened at the end of the sequencing, but up to now there is some evidence that what I explained you now can be true. So definitively in the baby, hopefully we can simply use blood or amniotic liquid or whatever you want for checking if the baby has this mutation.
Okay, thank you for Sri, Sylvia, and JP. We have another questions in the chat for Sri. Is there any simpler models compared to organotypic brain culture to study Parkinson's disease?
Okay, thank you for the questions. Yes, of course, there is a simpler model available. We have cell lines, of course. We can use also neuroblastoma, treated with the rotenone, kind of. But again, the problem is what we try with the ex vivo model is we try to maintain the heterogeneity of the cells, because in substantia nigra you have multiple cells, of course, besides the dopaminergic neuron, and we try to mimic this brain as much as possible. With the neuroblastoma, you will only have one type of cells, and it will mask the effect of the protection of UCB, I guess.
Thank you for the response, Sri. One quick question for you. So regarding the nanobubbles,
Yep.
do you think we can use that technology to deliver any other compound like anti-cancer or anti-inflammatory to use in different tissues or solid organs?
Yes. So far, what I know from this nanobubbles from developed in University of Turin. So actually, they developed this nanobubble as a vehicle or as a carrier for drug delivery purposes. So actually, we can use it and we can try to use it to test with any molecules or drug candidates that you want to use.
Okay, since there's no other questions in the chats and there are questions, I will invite Prof. Claudio Tiribelli for his comments or a response.
Thank you, Corey. And thank you, Nanning, for this excellent organization. I would like to make 2 points. Number one, to express my deep gratitude to all of you to get together this set of speakers and interested people to discuss something which is ongoing in 2 directions, and then actually highlighting the interaction we had with Sri and then with all of you between FIF and BRIN. But even more important, I think that we need to take home a message, which I would like really to stress. And I ask Professor Indy and Professor Farah to be the takeover and then the ambassadors to the Ministry of Health. Because in my opinion, there is a real need to make a study nationwide, taking as samples 2 or 3 centers for each big islands, north, south, east, and west, and then try to organize a survey study on the prevalence of severe neonatal jaundice by taking advantage of the point of care of measuring bilirubin we have. This is very simple, it's quite inexpensive, and it's very informative, because this study will give us 2 information. Number one, whether there is a local geographical difference, and I can tell you that the study we just finished in the Philippines show us very clearly that there are really large regional differences in the prevalence of severe neonatal jaundice. And I'm expecting that Indonesia, which is the country in the world with the largest genetic heterogeneity, may even more important. And number 2, as we have samples from blood of these babies who has undergo the study, we can use the blood stored in a cartridge to screen a chip when ready to understand whether we can really test in the field the idea Sylvia and AJP are running to prepare a chip in order to screen all the baby at no price for a danger in the development of severe neonatal jaundice. I think Indonesia is one of the best countries in the world able to do so. And I ask Professor Indy, Professor Para, and Sri, and any one of you to try to convince the government to do this study. the protocol is there. It's very easy to perform. In 3 or 4 months, we have all the data we have, and then will be important to include as a screening test for all the newborns a bilirubin measurement 24 hours, which actually predicts the development of severe neonatal jaundice. There's the clinical portion. In the parallel, we can save the samples, and then screen in terms of expression of genes, whether what JP is demonstrating in the rat is also true in a much more complicated animal model, which is the humans and particularly the newborns. So I convey this. I think that one of the major outcome of this seminar, the webinar, will be the starting at a sort of a national survey study on the prevalence of severe neonatal jaundice. In the parallel, the other deliverable which can take home this today is actually to strengthen even more the interaction between BRIN and Eichmann and all the research institutes in Indonesia and FIF. So I think we need to revitalize the flow of students and fellows from Indonesia to Trieste and back. And Cecil is actually the real ambassador in doing so, because she will be in Indonesia for 2 months, next 2 months. And she will discuss with BRIN people how to maximize the possibility to have the fellowship, which so far is rather, unflexible in terms of limit of age. But I'm confident that we can do this. But altogether, this means that the cooperation between BRIN and FIF is active, is productive, and is very with a very good potential for the future. Thank you very much.
Thank you for that remarks, Prof. Tribelli. So with that, I think I would like to close this discussion session. I would like to thank all the speakers again for their excellent talks and their insights and responses during the discussion. Our presenters today have focused on the neonatal hyperbilirubinemia and Parkinson's disease, describing the bilirubin neurotoxicity pathomechanisms and also novel therapeutic approach for severe neonatal hyperbilirubinemia, explaining the merit of translational research for neonatal jaundice and Parkinson's disease, and highlighting the neuroprotective effect of bilirubin in neurodegenerative diseases, particularly in Parkinson's disease. We hope that today's webinar will trigger more studies on bilirubin-associated diseases and initiate new potential research collaboration in Indonesia and also in overseas to further contribute to the global health efforts to reduce the morbidity and mortality associated with these conditions. Please join me again in thanking all our speakers, and thank you all for the participants for joining today's webinar. On that note, we will close this webinar session, and I will give the session back to our beloved MC. Thank you very much and have a good day.
Thank you, Corey, and thank you, Prof. Thank you, Sylvia, JP, and Sri for the lively discussion for today's webinar. And to end today's event, I would like to say thank you for all the speakers and the participants. And also, let's give a big applause to all of the speaker and the participant. I know it's online, so we cannot hear it. So big applause. And thank you once again for joining us today for this informative and engaging event. We hope you have gained valuable knowledge and insight from our esteemed speakers. We wish you all have a pleasant day and good day. Bye-bye. Ciao, ciao. Thank you.
Ciao, ciao. Thank you very much.
Thank you all.
Ciao, ciao. Thank you very much.
Ciao.
Bye-bye, Pencil.